It's my pleasure to introduce Dr. Christopher Hyman, who is a professor of preventative medicine at the Keck School of Medicine of USC and the Aflac Chair in Cancer Research. He also leads a cancer epidemiology program at the University of Southern California Norris Comprehensive Cancer Center and the Epidemiology and Genetics Division in the Department of Preventive Medicine. We have been extremely privileged and gratified by support from Robert F. Smith to create a polygenic risk score for men of African descent. Dr. Hyman is leading this effort by increasing the number of African American men whose germline genetics will be studied and whose risk will be assessed by a test that is coming along quite well at current time. The title of Dr. Hyman's presentation is A Polygenic Risk Score for Predicting Prostate Cancer in Men of African Descent. Dr. Hyman, it's our pleasure to hear about your research today.
Thank you, Howard, for that kind introduction. And thank you to the Prostate Cancer Foundation for the invitation to speak about our research in studying genetic susceptibility to prostate cancer and the work we've done in developing a polygenic risk score for predicting prostate cancer risk in men of African descent. I have no financial disclosure or conflict of interest with the material I will be presenting. So these are data from SEER, which highlight the well-known racial and ethnic disparities in prostate cancer incidence, with rates being 70% greater in African American men compared to men from other populations. African American men are also more likely to be diagnosed at a younger age and with more aggressive disease, and are twice as likely to die from prostate cancer than men from other racial and ethnic populations. With there being a limited number of established risk factors for prostate cancer, it has long been hypothesized that genetic factors might contribute to these differences. This is something we initially set out to test through a number of studies, starting with admixture mapping in 2006, which was work led by David Reich and Matt Friedman at Harvard. And in the study, we found a 4 megabase region on chromosome AQ24, where there was a significant excess of African genetic ancestry in African American men with prostate cancer. And within this admixture signal, we found 7 risk alleles for prostate cancer, located in 3 nearby regions in a gene desert, but in close proximity, as shown here, to MYC. A number of other studies around the same time also confirmed this region to be a very important susceptibility locus for prostate cancer in all populations. What was striking about this region was that almost all of the risk variants were found to be more common in men of African ancestry, as shown in blue here for just one of these risk alleles. And based on this observation, we constructed a polygenic risk model that accounts for the frequency and effect size of each of these 7 variants. And what we found was that when compared to white men, that the risk distribution was shifted to the right, to higher values for African American men, suggesting that AQ24 makes a greater contribution to prostate cancer risk in African American men compared to compared to white men or men from any other population. And this was really some of the first evidence to suggest that germline variation at this AQ24 region may be contributing to population differences in prostate cancer risk. So to continue this work, in 2007, we established the African Ancestry Prostate Cancer Consortium, which today includes 32 studies from North and South America, Africa, and Europe, and just over 12,000 prostate cancer cases and 12,000 controls. We've conducted a number of genome-wide association studies in this consortium and have observed that some of the strongest signals we see across the genome are with alleles that are only found in populations of African ancestry. And here are just 3 examples of such regions, and in total we have detected 5 risk variants with frequencies ranging from 2 to 10% in African ancestry populations that are not found in non-African populations. And these are findings that provide additional support for genetic factors contributing to population differences in prostate cancer risk, and also for the importance of conducting genetic studies in diverse populations. So in our studies in men of African ancestry, we've been able to confirm the very strong association with AQ24, which remains the strongest signal in the genome in any population. There are now around 15 independent risk variants for prostate cancer that have been discovered in this region. with one variant being a particularly important marker of risk for men of African ancestry, shown here. This is a germline variant that is only found in men of African ancestry with an allele frequency of 6%, meaning that 11% of men of African ancestry carry at least one allele, which conveys a twofold increased risk of developing prostate cancer. The frequency of this risk allele is greater in early onset and familial cases, which are the blue bars as shown here in this figure, with very high risks observed for both heterozygous and homozygous carriers with multiple affected family members, as shown to the right in orange and in gray. So we estimate this variant alone accounts for 10% of the familial risk of prostate cancer in men of African ancestry, which is double that estimated for HOXB13, mainly because this variant is 30 times more common in the population than HOXB13. And this raises the question whether patients and their relatives should be tested for this variant, as is recommended for men of European ancestry who carry the HOXB13 mutation. So far, I've discussed our work in men of African ancestry, but really most of what we have learned and most of the discoveries about genetic risk for prostate cancer has come out of large GWAS analyses in populations of European ancestry conducted within the Practical Consortium. And through the last year, just over 180 germline risk variants have been identified. Over the past few years, we have been working hard to increase the racial and ethnic diversity genetic studies of prostate cancer in this consortium to identify risk loci that may help to improve risk prediction across populations. And in our latest analysis, we combined genetic data for close to 240,000 men from these 4 population groups and identified an additional 86 novel risk variants for prostate cancer. And for at least a third of the previously known risk variants, we found stronger markers of risk through fine mapping, which we think is pointing us to variants that are more likely to be causal because of their panethnic effects on risk. And in total, we estimate these 169 risk variants now account for just over 40% of the familial risk of prostate cancer. So we've used these 269 markers to construct a polygenic risk score, as we did at AQ24. And again, this is a weighted measure that combines risk information across all variants for an individual. And what is shown here are the risks associated with different deciles of this risk score distribution. And so compared to men at average genetic risk in the population, which is defined here as the 40th to 60th percentile of this PRS distribution, we see that men in the 80th to 90th percentile have a 2.5-fold increase in risk, while men in the top decile to the far right have a 4 to 5-fold greater risk of developing prostate cancer. And you can see the risk is greater for men of European ancestry, which isn't too surprising, as most of the risk variants were initially discovered in studies in men of European ancestry. And we've been able to replicate the PRS associations into independent studies among men of European and African ancestry, as shown below here. And so there does appear to be relatively good transferability of the PRS across populations. But the performance is weaker, as shown here in some groups. And we have a number of studies underway, which I'll talk about in a minute, that are aimed at improving risk prediction across populations, particularly for men of African ancestry. So we've also looked at the PRS and analyses stratified by age. And here we found age to modify the risk for men in higher PRS categories. So this is shown here for men of European ancestry in the top left. So here, those in the top PRS decile category had an odds ratio of just over 4 for men older than 55, which is the dark green bar, whereas the odds ratio for men under the age of 55 was close to 7, which is the lighter green bar, as shown in the upper left. And this general pattern of effect modification was also observed for men of African ancestry, which is in the upper right panel, and also in each of the replication studies in men of European and African ancestry, which are the 2 lower panels. And so in general, cancer diagnosed at a younger age typically indicates a genetic influence, and this does seem to be the case here for prostate cancer, with the PRS having a greater impact on prostate cancer risk for earlier versus later onset disease. So an important question with respect to the potential clinical utility of the PRS is whether it can differentiate risk of aggressive versus non-aggressive prostate cancer. So as shown here in light green is the percentage of aggressive high-risk cases within each PRS decile category. And here, aggressive disease is defined as stage T3 or T4, Gleason of 8 or higher, metastatic or lethal prostate cancer. And you can see that these proportions are identical to those observed for non-aggressive disease within these PRS deciles, which are the bars in darker green. And this is also the case for men of African ancestry, as shown below here in red. And so for the current PRS, we do not think it can be used to differentiate a man's risk of developing aggressive versus non-aggressive disease. However, what is quite noticeable from these figures is that a sizable fraction of all cases occur within a relatively small fraction of the population. So here, around 50% of all cases, including 50% of all of the high-risk aggressive cases, occur among the 20% of the population at highest genetic risk. And so while the PRS cannot predict who is more or less likely to develop aggressive disease, it can be used to define a smaller subset of the population in which a substantial fraction of aggressive cases will occur. And like we did early on at AQ24, we have also compared these new PRS distributions across populations. And here we find the distribution to be higher for men of African ancestry, in red compared to men of European ancestry in green. And the shift to the right for men of African ancestry is driven, again, by AQ24, but also many other risk variants that are more common in men of African ancestry. It places a larger fraction of the African ancestry population at higher risk. And so we also can compare the means of these PRS distributions between populations. And here we estimate that the PRS conveys around a twofold greater risk of prostate cancer for men of African ancestry compared to men of European ancestry. And certainly we have more work ahead of us to confirm these findings, but so far, the data do suggest that the known risk variants for prostate cancer may account for a substantial fraction of the greater incidence of prostate cancer that is observed in men of African ancestry. But what we also want to know is, can the PRS tell us something about a man's lifetime risk of developing prostate cancer? And unfortunately, prospective studies with genetic data across populations doesn't currently exist, but we can estimate absolute risk for men in each PRS category, which we have done using incidence and mortality data from SEER for whites and African Americans. And each of these different color lines here are absolute risks for different categories of the polygenic risk score over a man's lifetime. And just for reference, the average population risk is the 40 to 60th PRS category, which is this black line in each of the figures, with a average lifetime absolute risk being around 12%. So the lines here at the top in red are for men in the top 1% of the PRS distribution, who are estimated to have a lifetime risk of over 60% for whites and 50% for African American men, with men in the top 10% of the risk score, which is the orange line here, having lifetime risks of around 40% for both white and African American men. And these estimates need to be confirmed in future prospective studies, but do infer that the PRS can be used to identify subgroups of men across populations who have very high or very low lifetime risks of developing prostate cancer. This slide here really underscores the importance of diversity in our genetic studies. And so the pie charts shown here below are the overall number and the proportion of the different ancestry groups in our studies over the last few years. And this figure above here illustrates the impact this has made not only on the study power and the success of these studies to discover new risk variants, but also on the improvement in the performance of the polygenic risk score. And this is mainly because the greater diversity of the sample allows us to find variants that are more likely to have pan-ethnic effects on risk. And so as we move forward, our goal here is to improve the performance of the PRS across populations for both overall prostate cancer, but also for aggressive disease. And to do so, it's going to be critical to continue to increase the diversity of these studies and also to integrate the common variants from GWAS with pathogenic mutations and other rare variants identified from ongoing large-scale sequencing studies. And we hope to accomplish this through collaboration with existing biobanks and new recruitment efforts. And over the next 2 to 3 years, we expect to reach probably over 200,000 multi-ethnic cases, including 50,000 with whole exome sequence data. And this sample will include a considerably larger proportion of men from the non-European ancestry populations. So one such study that we expect to make a major contribution to this work is the RESPOND study. So RESPOND is a nationwide effort to study social and biological factors that contribute to aggressive prostate cancer in African American men. And we are currently in the process of recruiting a cohort of 10,000 African American men with prostate cancer through the cancer registries in 8 states, as shown here. And we're really enormously grateful to Robert Smith, who has again joined forces with PCF to address prostate cancer health disparities. And with his support, we hope to enhance recruitment activities in RESPOND and accelerate further optimization of the polygenic prostate cancer test for men of African ancestry. And just to end, a major question that remains is the clinical utility of the PRS. And as I've shown you, the PRS can be used to define 10% of men with lifetime risks that range from 40 to as high as 60% for the top 1% of the population. And these estimates are even higher for men who also have a family history of prostate cancer. So the PRS is also likely to augment risk for pathogenic mutation carriers, which raises the question whether the PRS should be incorporated in genetic testing, particularly for men with a family history. Another potential clinical utility of the PRS is for risk stratified screening. And even though the PRS can't differentiate a man's risk of aggressive versus non-aggressive disease, it can reduce the pool of men who are more likely to develop aggressive disease, with 50% of lethal cases occurring among 20% of men at highest genetic risk. And a risk stratified screening approach is also expected to reduce the number of biopsies and overdiagnosis and overtreatment of indolent disease. And this is definitely something that needs to be examined in prospective studies alongside other risk factors such as early life PSA. And so I think this is really where this work is heading, and hopefully a prospective and multi-ethnic PSA screening trial will be considered that incorporates everything we know and will continue to learn about a man's underlying risk of prostate cancer. And to end, I'd like to acknowledge all of my collaborators in the AAPC consortium, those at USC, and special thanks to all the funding agencies. So NCI, NIMHD, NHGRI, Robert Smith, and the Prostate Cancer Foundation. Thank you. Great. So one of the first questions that was asked is, what is the plan in the near term to increase accrual, validation, and partnerships? So we are currently working with the Million Veterans Program right now. As many people are aware, they have genome-wide association data on more than 500,000. Well, hopefully it will reach 1 million in the near future. And there are 50 to 70,000 prostate cancer cases within that data set. And we're currently working with them now to validate, further validate the polygenic risk score across populations. They have substantial numbers of men of African ancestry and Hispanics. And they will also be included in the next round of multi-ethnic or trans-ethnic meta-analyses for prostate cancer to substantially increase the power to identify novel risk variants for overall prostate cancer, but also for aggressive disease. We're also working on partnerships with the MADCAP Consortium, which is an international study led by Tim Rebick at Harvard, which is currently leading a large genome-wide association study of prostate cancer across different countries in Africa. And our goal, probably in the next 2 to 3 years, will be to construct a genetic data set that will have over 35,000 prostate cancer cases of men of African ancestry and 70,000 prostate cancer controls. And I think integral to this as well will be the study that I mentioned, which is the RESPOND study, which will contribute on upwards of 10,000 prostate cancer cases among men of African ancestry. So another question has been the accrual of African American men to clinical trials has always been a challenge. And what are our plans to actually increase accrual to trials and of these men in these studies? And I think this has really been the biggest challenge that we have faced. I'm speaking now of the RESPOND study, has been recruitment. As recruiting 10,000 men of African ancestry across the U.S. is no easy task. And in getting the study up and running, we conducted focus groups of African American prostate cancer patients at each of the different recruitment sites across the U.S. to get their views about how we build trust and credibility with the African American community so that we're successful in these recruitment goals. And these men provided very valuable information about their concerns regarding participation that we really took to heart and made sure that we addressed and articulated when we were asking for their participation, such as making it clear the goals and the intention of the research, what the real benefits are for them and for their families, and the need for transparency of the research and confidentiality of the data. They've also provided suggestions as to how to build credibility and trust with the African American community, getting buy-in from African American community leaders and organizations. And this is information, we've all integrated this information into all of our recruitment materials and plans for publicity to increase, hopefully, the likelihood of recruitment in the RESPOND study. Another question is, what are our plans to work with the VAs to enhance accrual in these studies? And as I mentioned, we're currently working with the Million Veterans Program, but we're also working with the Prostate Cancer Foundation, the VA, Centers of Excellence, and having dialogue with those centers about the potential of them becoming a site for the RESPOND study to help increase accrual and inclusion of veterans in RESPOND to study the social stressors, to study germline genetics, to study tumor biology as it relates to both prostate cancer incidence, but prostate cancer aggressiveness and outcomes. And these are discussions that are currently ongoing, and we're hopeful that the VA, we can work more closely with the VA in the future on these efforts. Another question is related to the polygenic risk score. As presented, it does not differentiate risk between low and high risk prostate cancer. And really, what is the biology that one thinks is behind this? It has, we've, unlike breast cancer, where there are specific variants that are associated with ER-negative disease and more aggressive forms of breast cancer, for prostate cancer, the common genetic variants that we've identified do not differentiate risk, which I think the hypothesis here is that they're involved in the very early stages of prostate cancer development. We know that there are rare variants and the more highly penetrant variants, DNA repair genes and others, that are involved in more aggressive phenotypes of prostate cancer. But it appears the common variants are most likely just involved in the early stages of the development. It also raises the question whether what are the non-genetic factors that are contributing to prostate cancer aggressiveness and progression. I think we need more work on this, particularly for men of African ancestry, looking at diet and other environmental factors that could be important in developing aggressive prostate cancer.